All right, so photosynthesis. Here is the chemical equation of photosynthesis. Get my pointer. Oh, wait. So we have carbon dioxide, water, and light as reactants going in, and sugar and oxygen are going to be the products that are at the end of this reaction. Oops. All right. So quick note about chemistry. Living things create bonds to store energy. So when we're doing our anabolic reactions, we're bringing atoms and molecules together to create bonds. We're making a place for that energy to be stored. But when we break those bonds, we're releasing energy. So in our catabolic reactions, when we break bonds, we're releasing that energy that was stored in the bond originally. So let's look at something like glucose. So glucose is our monomer for carbohydrates. Why do you think carbs are such good energy? Well, let's look at all of these bonds. Carbs are made up of glucose molecules. And there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 bonds. 16 bonds in one little tiny glucose molecule. That's a lot of stored energy. So that's why glucose is like primo food. Lots of bonds to break, lots of energy to get from that. All right, so then there's also these energy molecules you may have heard of. ADP, adenosine diphosphate. So using our roots here, di means two. There are two phosphate groups here. And ATP, adenosine triphosphate. Tri meaning three, again our roots. And there are three phosphate groups attached to this adenosine molecule. So we got ADP, which has two, there's only two bonds there, and ATP, which has three bonds. So naturally, the ATP will be a little bit more energy than ADP. So we want ATP, we can still use ADP, but ATP triphosphate is the one. All right, so photosynthesis generally, like big picture photosynthesis, it has two parts. It has light dependent or light reactions, so reactions that happen that need and require light. And there's another half about light independent reactions. So these reactions, it's also called the Calvin cycle. We're going to talk about that tomorrow. They don't need light to happen. So we have light dependent, light independent. One needs light, one doesn't. And when we're talking about our chemical reaction, if you remember, we need carbon dioxide to go in, and our waste product is going to be oxygen. Well, these are gases that need to go in and out of the plant. How does that happen? Well, plants have like leaves, stems, things like that, and they have like these little pores. Like our skin has pores, it helps things come in and out. Plants also have pores, but they're called stomata, and they can like open and close and let gases in and out. So when they need some carbon dioxide, they'll open, harvest some carbon dioxide. When they're doing their photosynthesis, they're going to have oxygen as waste, and that will need to leave the plant as well. So the stomata kind of regulate the gas exchange between the inside and the outside of the plant. All right, let's get into it. Light dependent reactions. So first thing, we have to look at our chloroplasts. Not very big, but still bigger picture. We're going to say we have our plant cell, right? And in our plant cell, we have our organelles. One of those organelles in a plant cell is something called a chloroplast. This is a chloroplast. So we're starting not very big, but still big, getting a little bit smaller to the chloroplast. And then we're going to get even smaller looking at the pieces of this chloroplast. So chloroplasts have two membranes, an outer and an inner. They have things called thylakoids. So we see these like stacks of discs. One of those discs is called a thylakoid. And a stack of those thylakoids is called a granum. These granum are kind of situated inside the chloroplast, but they're not just like hanging out in a vacuum with no air. They're not hanging out in there with air. What they're in is something called stroma. It's like this aqueous solution. It's water-based, and like it's kind of like the cytoplasm in a cell. It kind of makes sure everything is cozy and happy. They're not like moving around and flopping and getting shaken up. So the stroma is like this aqueous space that things are chilling in, all right? So our light reactions, here is the chemical components. We got water and light, and they're going to react and create ATP, our energy, and something called NADPH, we're going to talk about what that means, and oxygen, our O2. So our three important things that it does is it produces ATP, NADPH, and releases O2 as waste. So it's getting rid of that oxygen. It doesn't want oxygen. Plants produce oxygen as a waste. So what's the purpose? We just talked about it. We're making ATP, NADPH, and we're getting rid of oxygen. So there are 4 parts, 4 steps. We're going to start, this is counterintuitive, but bear with me. We're going to start with something called photosystem 2. I know, starting with 2, kind of weird. Then we're going to the electron transport chain, or it's often called the ETC. We're going to move electrons to our photosystem 1, and then we're going to end with ATP synthesis. We're going to make that ATP, or energy. So this part is kind of confusing because, like I said, we're starting with photosystem 2. It's named this way because scientists discovered photosystem 1 first and didn't realize that stuff came before that. And then they did more research and they found photosystem 2, and they're like, well, we already named one of them. I guess this one's going to have to be photosystem 2. I'm not in charge. I don't name these things. We're just going to have to deal with it. We have to remember that photosystem 2 comes first, and then photosystem 1. So here's a general overview of what's going on. So here's our chloroplast, medium picture. We're going to get small, even smaller, and we're going to have light comes into our photosystem 2. An electron travels through some enzymes to photosystem 1. An electron travels through a couple more enzymes, and then the last step will be that ATP synthesis, making ATP. So let's get even smaller. Let's focus on the first step, photosystem 2. So in photosystem 2, it has a special pigment. Pigment in plants is chlorophyll. It's something that the light will react with to, give us, to harvest that light, those photons. In photosystem 2, that pigment is called P680. You don't have to know that. You just need to know that it has a different pigment than photosystem 1. That's their main difference, is this different pigment names. So photosystem 2, light comes in, a photon hits that pigment. In this image, those pigment molecules are these dark green dots. Those dark green dots get a little excited, and they're going to transfer that excited energy to an electron. That electron, because those pigment molecules were excited, it's going to get excited. It's like getting charged up. That electron is going to, thanks for letting me know, Henry. That electron is going to then do a couple of things. Its main job is to go on to the next step, the electron transport chain. But it also starts a new process with an enzyme. So there's an enzyme in photosystem 2, and what it's going to do is it's going to take a water molecule. We and plants, most living organisms, are water-based. We have water in and out and around all of our stuff, cells, thylakoids, all that. So there's water inside this thylakoid, and that enzyme is going to take a water molecule, or rather 2, and it's going to break them apart. We're going to get an oxygen and 2 hydrogens. But in order to make that O2 molecule, we need to break apart 2 water molecules. So this enzyme is going to break apart 2 water molecules so it can make an O2 molecule that can be waste. It's going to get sent out of this system, out of the plant. We don't need that O2. But we're going to keep those hydrogen ions. Those hydrogen ions are going to be super important in a little bit. So once again, light comes in, hits these P680 pigment molecules. Those get excited. They tap into this electron. This electron sets off a reaction with an enzyme. That enzyme is going to split water molecules, so it has O2 that leaves. Bye-bye, we don't want you. And some hydrogen ions, which we want to keep inside that thylakoid. Okay, photosystem 2. summary. I know it sounds repetitive, but sometimes it just takes repetition with this complicated stuff. Here's the little sun shining down, hitting those pigments. Those pigments transfer energy to electrons, which are energy in their own sense. And the main job of the electron is to travel down the next step, the electron transport chain, but it's also triggering an enzyme, which will split a water into oxygen and hydrogen ions, excuse me. And those hydrogen ions are going to create a concentration gradient. Hey, some vocab from last unit, concentration gradient. We're gonna have a lot of hydrogen ions inside the thylakoid than outside. All right, so we're moving on. We're focusing on this electron that got really excited. Some molecules like to play a game of hot potato. So we're gonna take this electron and go, no, you have it, no, you have it, no, you have it. And we're gonna go down this thing called the electron transport chain. This is a diagram. Obviously there aren't little people inside thylakoids working, but it's sort of like a little way to look at how this electron transport chain goes. So here's at the bottom, we're going to start with a photon from light, and that sunlight is going to excite this electron, and the electron is going into photosystem 2. Photosystem 2 is going to push it down this electron transport chain to photosystem 1. It's going to create some NADPH after it receives some more sunlight. We're going to talk about why. And lastly, it'll create this ATP. So this electron is just moving through what's called a chain. And so here we go. Here's our photosystem 2. This electron gets excited, triggers that enzyme to split apart the water, but then its main job, it's going to go through a couple enzymes. I gave you the names of the enzymes. You don't need to know them. I can't even pronounce them. I don't really care what they're called. Just know that these enzymes, their main job is to continue to contribute to that H+ concentration gradient. They're going to take a little bit of that energy from the electron and use that for active transport. Vocab from last unit again. Active transporting those hydrogen ions from the stroma, the outside of the thylakoid, to the inside. We're going to keep pulling those H+ ions in. We need this high concentration inside the thylakoid. So as these enzymes are transporting this electron through, they're taking a little bit of energy, using it to pull in hydrogen ions. And then this electron, as it's traveling through these enzymes, will end up here at photosystem 1. Okay, those enzymes stole a little bit of its energy. So we're going to use light again to excite these pigment molecules in photosystem 1. These pigment molecules are called P700. Again, you don't need to know that, just know that they're different. The last one was P680, this one's P700. Just a very little itty bitty tiny difference. Same thing essentially is happening in this photosystem. The light is exciting these pigment molecules. The pigment molecules are transferring that excitement energy to the electron. And then the electron is going to move again, this time through a much shorter electron chain. So here, light hitting those pigment molecules, exciting the electron, going through a couple more enzymes. Now these enzymes, they're going to take that electron that was passed to them. They're going to reach into the stroma, pick up an H+, and that NADP+, that was a product of our last reaction that we're going to talk about tomorrow, but will be a reactant here. And it's going to smush them together so that we have what's called an NADPH. This NADPH will travel to the reaction we're going to cover tomorrow. But what does this mean, NADPH? This doesn't have the P in it because these are the things that are in cellular respiration, but it's the same concept, except there's a P in there. So here we're going to have our NADPH+, and it's taking those electrons from the electron transport chain. It's taking a hydrogen ion from the stroma, and they're smushing it together. And it's going to carry these electrons. This is called an electron carrier molecule. Its sole job is to carry electrons from one place to another. So it's going to take these electrons, carry it over to the reaction we're going to talk about tomorrow, the light-independent reaction. So that's all that's happening with this NADP+ and NADPH. It's tacking on a hydrogen ion to trap in some electrons, move them on to a different reaction. Photosystem 1 summary. Light energy, we're adding more light again, is absorbed by a pigment. That NADP+ and H+ from the stroma are going to smush together with an electron to form NADPH. Lots of letters, I know, I apologize, but it's much easier than saying the entire whole molecule and trying to remember that. So just know if there's an H on there, on the end, there's no plus, it's going to be an electron carrier. All right, the last step, ATP synthesis, the most important part. We want to get that energy, that ATP. So this is where that concentration gradient comes in, super duper important. So inside the thylakoid, we have been building all of this H+ ions, right? We had the splitting of water that added H+ ions. We had those enzymes at the first electron transport chain that were pumping in those H+ ions. Now we have all of this concentration on the inside of our thylakoid. Well, what we know from our last unit, talking about concentration gradients and how they interact with membranes, those H+ ions want to leave the thylakoid. They want to balance out that gradient, right? And their only way out right now is through a new enzyme called ATP synthase. Enzymes are often named after the jobs that they do. So this one kind of gives it away. It's synthesizing, it's creating, synthesizing synthase, ATP, right there in the name. So it's using this gradient to channel in those H+ hydrogen ions so that they're going through this enzyme and it's powering the production of the ATP. So we received adenosine diphosphate, DP, from, again, it's a product of the reaction we're going to go over tomorrow, and a phosphate group, and it's going to stick them together as these hydrogen ions are pumping through it. A good analogy is like, it's sort of like a turbine. So when you think about a dam, and the water is falling and turning these turbines, and it's producing electricity. A similar thing is happening here. Those H+ ions are going through this enzyme, creating this buildup of energy because they're trying to get through, and it's essentially creating a little bit of energy to create these ATP molecules. Stick that last phosphate group onto the ADP, the diphosphate. So this image is upside down, so don't get too confused. So up here we have our thylakoid, down here we have our stroma. It's opposite the way the last one looked. But here we have all of these H+ ions, and they want to leave the thylakoid. They are crowded. This concentration is too much inside. So they're gonna go through the ATP synthase enzyme, and as that's happening, the ATP synthase is using that to create ATP. All right, that is all we're gonna cover today. I know I talked for a while, but again, this stuff is really complicated. If you have questions, please stick around, come to office hours, send me an email. If you feel okay, there is this worksheet. It's group, but you can start this top part if you feel like you want to do that. You're just labeling parts on this model. Tomorrow we're going to talk about this, the light independent reaction, and then there's a CER to do when we've covered both of those. So if you want to start on this, great. If you want to wait, also fine.